And welcome back to this new video of this chapter, outbreeding devices. Majority of flowering plants produce hermaphrodite flowers, and pollen grains are likely to come in contact with the stigma of the same flower. Continued self-pollination results in inbreeding depressions. Flowering plants have developed many devices to discourage self-pollinations and to encourage cross-pollination. In some species, pollen release and stigma receptivity are not synchronized. Either the pollen is released before the stigma become receptive, or stigma become receptive much before the release of pollen. In some other species, the anther and the stigma are placed at different positions so that the pollen cannot come in contact with the stigma of the same flower. Both these devices prevents orthogamy. The third device is to prevent inbreeding is self-incompatibility. This is genetic mechanism and prevents self-pollen from the pollen flower or other flower of the same plant from fertilizing the ovule by inhibiting flower pollen germination or pollen tube growth in pistil. Another device Another device to prevent self-pollination is the production of unisexual flower. If both male and female are present on the same plant, such as castor, maize, which is monoecious, it prevents orthogamy but not geitonogamy. In several species, such as papaya, male and female flower are present on different plants. That is, each plant is either male or female, dioic. Such conditions prevent both orthogamy and geitonogamy. Pollen pistil interaction. Pollen does not guarantee the transfer of the right type of pollen, compatible pollen of the same species as the stigma. Often, pollen of the wrong type is either from the other species or from the same species. If it is self-incompatible, also land on the stigma. The pistil has the ability to recognize the pollen, whether it is the right type or the wrong type. If it is the right type, the pollen sac accept the pollen, the pistil accepts the pollen and promotes post pollination events. That leads to fertilization. If the pollen is of wrong time, the pistil rejects the pollen by preventing pollen germination on the stigma or the pollen tube growth in the style. The ability of the pistil to recognize the pollen followed by the acceptance or rejections is the result of continuous dialogues between the pollen and the pistil. This dialogue is mediated by chemical component of the pollen pistil interaction with those of the pistil. It is only in recent years that botanists have been able to identify some of the pollen and pistil component and the interaction leading to the recognition followed by active acceptance or rejection. As mentioned earlier, following compatible pollinations, the pollen grain germinates on the stigma to produce a pollen tube through one of the germ pole. The content of the pollen grain moves into pollen tube. Pollen tube groups grows through the tissue of the stigma and style reaches the ovary. You may recall that in some plants, pollen grain are sets at two cell condition, a vegetative and a generative cell. In such plants, the generative divides and forms two male gametes during the growth of pollen tube in the stigma. In plant which said pollen at three cell structure, pollen tubes carry the two main structures from the beginning. Pollen tube, after reaching the ovary, enters the ovule through the micropyle and enters one of the synergids through the filiform apparatus. Many recent studies have shown that filiform apparatus present at the micropylar end of the synergids guides the entry of the pollen tube. All these events from pollen deposition on the stigma until pollen tube enter the ovule are together referred as pollen pistil interaction. As pointed out earlier, the pollen pistil interaction is a dynamic process. involving pollen recognition followed by promotions or innovation of the pollen. The knowledge gained in this area would help in plant breeder in manipulating pollen pistil interaction even in incompatible incompatible pollination to get desired hybrid. You can easily study pollen germination by dusting some pollen on off flowers such as pea, chickpea, balsam, vinca on a glass slide containing a drop of sugar solution about 10%. After about 15 to 30 minutes, you will observe that the slide under the low power lens of the microscope, you are likely to see pollen tubes coming out of the pollen grains. As you shall learn in this chapter on plant breeding, chapter 9, a breeder interested in crossing different species and often genera to combine desirable characters to produce commercially superior varieties. Artificial hybridization is one of the major approach of crop improvement program. In such crossing experiment, it is important to make sure that only the desired pollen grains are used for pollination and the stigma is protected from the contamination. This is achieved by emasculation and the bagging technique. If the female plants bear bisexual flower, removal of anther from the flower bud before the anther desiccates using a pair of forceps is necessary. This step is referred as emasculation. Emasculated flower had to be covered with a bag of suitable size, generally made up of butter paper, to prevent contaminations of its stigma with unwanted pollen. This process is called bagging. When the stigma when the stigma of bagged flower attains receptivity, mature pollen grain collected from anther of the male parent are dusted on the stigma, and the flower are rebagged and the fruit allowed to develop. If the female parent produced unisexual flower, there is no need for emasculation. The female flower buds are bagged before the flower are open. When the stigma became receptive, pollination is carried out using the desired pollen and flower is rebagged. Double fertilization is very important. This is very important in angiosperms. After entering one of the synergids, the pollen tube releases the two male gametes into the cytoplasm of the synergid. One male gamete moves towards the egg and fuses with the nucleus, thus completing syngamy. This results in the formation of diploid cells. The other male gamete moves towards two polar nuclei located in the central cell and fuses with them to produce a triploid primary endosperm cell, which is known as pen. As this involves the fusion of three haploid nucleus, it is termed as triple fusion. Since two two type of fusion syngamy and the triple fusion takes place in the embryo sac, the phenomenon is termed as double fertilization, and this unique to flowering plant. The central cell after triple fusion becomes the primary endosperm cell and develops into the endosperm, while zygote develops into embryo. This is the figure of embryo. Post fertilization structure and events. Following double fertilization, events of endosperm and the embryo development, maturation of ovules into seeds, ovary into fruit are collectively termed as post-fertilization events. Endosperm. Endosperm development precedes embryo development. Why? The primary endosperm cell divides repeatedly and forms a triploid endosperm tissue. The cells of this tissue are filled with reserve food materials and are used for the nutrition of developing embryo. In most common type of endosperm development, the pin undergoes successive nuclear division to give rise to a free nuclei. This stage is known as prenuclear endosperm. Subsequently, cell wall formation occurs and the endosperm becomes cellular. The number of free nuclei we form before cellularization varies greatly. The coconut butter from tender coconut that you are familiar with is nothing but free nuclear endosperm made up of thousands of nuclei, and the surrounding white kernel is called cellular endosperm. These are plumule. This is plumule, two of the cotyledons, hypocotyle. This is also known as this will known as epicotyle. This will term as epicotyle. Endosperm may be either be completely consumed by the developing embryo, example pea, ground beans, before the seed maturation, or it may persist in the mature seed. These will consume totally and persist in castor and coconut and be used up during seed germination. Split open some seeds of castor beans, walnuts, fruits of coconuts, and look for the endosperm in each case. Find out whether the endosperm is persistent in cereal, wheat, rice, and maize. This is scutellum monocot. It is a dicot embryo. These two sides are coleoptiles. This is shoot apex. This is epiblast. The inner one is radical, and the middle one is root cap, and the outer one is coleorhiza. Embryo. Embryo develops at the micropylar end of the embryo sac where the zygote is situated. Most zygotes divide only after certain amount of endosperm is formed. This is an adaptation to provide assured nutrition to the developing embryo. How? How the seeds differ greatly. The early stage of the embryo development are similar in both monocotyledon and dicotyledons. Depicts the stages of embryogeny in dicotyledon embryo. The zygote gives rise to the proembryo, subsequently to the globulars, heart shape, and mature embryo. A typical dicotyledonous embryo consists of embryonal axis, two cotyledons. The portion of umbilical axis above the level of cotyledons is the epicotyle, which terminates with the plumule or stem tip. The cylindrical below level of the cotyle is known as hypocotyle. That terminates at its lower end, known as radical and root tip. The root tip is covered with root cap. Embryo of monocotyledon possesses only one cotyledon. In the grass family, the cotyledon is known as scutellum, which is known as scutellum. That is situated towards one lateral side of the embryonal axis. At its lower end, embryonal axis has the radical and root cap enclosed in an undifferentiated sheath called coleorhiza. The portion of the embryonal axis above the level of attachment of a scutellum is the epicotyle. Epicotyle has a shoot apex and few leaf primordia enclosed in a hollow follicular structure known as coleoptiles. Seed. In angiosperm, the seed is the final product of the sexual reproduction. It is often described as a fertilized ovule. Seed is represented as fertilized ovule. Seeds are formed inside fruits. A seed typically consists of seed coat, cotyledons, and an embryo axis. The seed is consist of seed coat, cotyledons, embryo axis. Embryo axis. The cotyledons of the embryo are simple structures, generally thick and swollen during storage of food reserve. Mature seeds may be non-albuminous or ex-albuminous. Non-albuminous seeds that have no residual endosperms at is completely consumed during the non-albuminous seeds have no residual endosperm assets completed during the embryo development. Pea and groundnut have non-albuminous. Albuminous seed retain a part of endosperm and it is not completely used up during embryo development, which is wheat, maize, barley, castor. Occasionally, in some seeds such as black pepper and beet, remnants of nucellus is also processed. This is also known as perisperm. Integument of ovules. Integuments of ovules harden as tough protective seed coats. The micropyle region as a small pore is the seed coat in the seed coat. Once again, integument of ovules hardens as tough protective seed coats. The micropyle remains as a pore in the seed coat. This facilitates the entry of oxygen and water into the seed during germination. As the seed matures, water content is reduced and seed becomes relatively dry, 13 to 15%, 10 to 15% moisture by mass. The general metabolic activity of the embryo is slowed down. The embryo may enter a state of inactivity called dormancy, of or if favorable conditions are available, adequate moisture, oxygen, and suitable temperature, they germinate. As ovule matures into seeds, ovary develop into fruit. That means the transformation of ovule into seed and ovary into fruit proceeds simultaneously. The walls of the ovary develop into the ball of the fruits called pericarp. The ball of the ovary develops into the ball of the fruit known as pericarp. The fruit may be fleshy as in guava, orange, mango, etc., or may be dry as in groundnut and mustard. Many fruits have evolved mechanism of dispersal of seeds. Recall the classification of fruits and their dispersal mechanism that you have studied in an earlier class. Is there any relationship between the number of ovules in an ovary and the number of seeds? Yes, ovules forms the seeds, so there must be a relationship. In most plants, by the time of fruit development from the ovary, other floral parts degenerates and Others fall off. However, in few species such as apple, strawberry, cashew, the thalamus also contribute to the fruit formation. Such fruits are known as false fruits. Most fruits, however, develop only from the ovary is known as true fruits. Although in most of the species, fruits are the results of fertilization, there are few examples in which fruit develop without fertilization. Such is known as parthenocarpic fruit. Parthenocarpic fruit. Banana is one of such example. Parthenocarpy can be induced through the application of growth hormones and such plants and such fruits are seedless. Seeds offer several advantages to angiosperm. Firstly, since reproductive processes such as pollination and fertilizations are independent of water, seed formation is more dependable. Also, seeds have better adaptive strategy for dispersal to new habitat and help the species to colonize in another area. As they have sufficient food reserves, young seedlings are nourished until they are capable of photosynthesis on their own. The hard seed coat provides the protection to the young embryo. Being the product of sexual reproduction, they generate a new genetic combination leading to variation. Seed is the basis of our agriculture. Rehydration and dormancy of the mature seeds are crucial for the storage of seeds which can be used as food throughout the year and can be raised crops for the next season. Can you imagine agriculture in the absence of seeds or in the presence of seed which germinate straight away soon after formation and cannot be stored? How long do seeds remain alive after they are dispersed? This period again vary greatly. In a few species, the seed loses viability within a few months. Seeds have a large number of species lives for several years. Some seeds can live alive for hundreds of years. Several are record of very old and viable seeds. The oldest is the Lupinus arcticus, excavated from the Arctic tundra. The seed germinated and flowers after an estimated record of 10,000 years of dormancy. A recent record of 2,000-year viable seeds of the date palm Phoenix dactylifera, discovered during the archaeological excavation of King Herod's place in near the Dead Sea. After completing a brief account of sexual reproduction of flowering plant, it would be worth attempting to comprehend the enormous reproductive capacity of some flowering plants by asking the following questions. How many eggs are present in an embryo sac? How many embryo sacs are present in ovule? How many ovules are present in the ovary? Can you think of some plants in which fruits contains very large number of seeds? Orchid fruits are one such category and each fruit contains a thousand of tiny seeds. Similar in the case in fruits of some parasitic species such as Orobanchi and Striga. Have you seen a teeny seed of ficus? How large is the tree of ficus developed from the teeny seeds? Apomixes and polyembryony. Although seeds in general are the products of fertilization, a few flowering plants such as some species of Asteraceae grasses have evolved a special mechanism to produce seeds without fertilization called apomixes. What is the fruit production without fertilization called? Thus, apomixis is a form of sexual reproduction that mimics sexual reproduction. There are several ways of development of apomatic seeds. In some species, the diploid egg cells is formed without reduction division and develops into the embryo without fertilization. More often, as in many citrus and mango varieties, some of the nuclear cells surrounding the embryo sac start dividing, protrude into the embryo sac and develop into the embryo. In some species, each ovule contains many embryos occurring or more than one embryo in a seed is referred as polyembryony. Take out some seeds of orange and squeeze them. Observe that many embryo of different seeds and same from each seed contain the known number of embryo in each seed. What would be the genetic nature of apomatic embryo? Can they be called clones? Hybrid varieties of several of our food and vegetable crops are being extensively cultivated. Cultivation of hybrids has tremendously increased productivity. One of the problem of hybrid is that hybrid seeds have been produced every year. If the seeds collected from hybrids are sown, the plants in the progeny will segregate and do not maintain hybrid characters. Production of hybrid seed is costly, hence the cost of hybrid seeds become too expensive for the farmer. If these hybrids are made in apomatic, there is no segregation of character in the hybrid progeny. Then the farmers can keep on using the hybrid seeds to raise new crops year after year and has does not have to buy new hybrid seeds every year because of the importance of apomixes in hybrid seed industry. Active research is going on in many laboratories around the world to understand the genetics of apomixes.